Hey guys, welcome back. Today I'm going to be walking through how I count cells. So here we are again in my cell culture hood. First I have a suspension of cells that I'm going to just mix up quickly before I grab a very small amount to add to another tube. The second tube contains trypan blue, which is that really dark solution that you see on the left-hand side of the hood. This actually stains cells that are dead versus the ones that are alive, which I'll show you what that looks like on the cell counter later. This is one of the slides that we're going to be using to inject some of our cell solution into. I'm just going to be picking up a small amount of that solution that was mixed with the trypan blue and injecting it into a little tiny hole where it can spread out over the entirety of that plastic slide. I'm going to repeat this for 2 samples per each of these plastic slides. And I actually had a bunch of samples you can see here, but I completed that for all of those. This is an automated cell counter that I'm going to put the plastic slide into. And here is actually an image of a very dense sample. All of those little dots circled in green are cells that are alive. The red ones are dead cells where the trypan blue was able to get into the cell, meaning its membrane is no longer intact and the cell is considered dead. There are a couple of things that you can do with the numbers that the cell counter records, such as calculate dilutions for replating out cells. You can also gate cells based off of their morphology, which I'm showing you here. And lastly, in just a second, I'm going to show you what it looks like when your cells are a lot less dense than you expected. And I'll actually be using all of these cell counts to help plate out cells for another experiment that will be coming up in another video.